Dear friends, once again it's hearty and warm welcome. Friends, in this lecture today we are going to learn major topic, that is translation. All of you are know that the actual process of protein synthesis involves two major steps such as transcription, and it is followed by translation. We have already studied that the basic genetic information necessary for the process of protein synthesis is present on DNA, known as genetic code. This information from DNA is inscribed, printed on messenger RNA. This process is called as the transcription. Now this messenger RNA carries that information in the form of codons up to the ribosome, where actually protein is synthesized. And therefore the translation, it is the second step in the process of protein synthesis, in which the polypeptide molecule or protein molecule is synthesized according to the codons present on messenger RNA in presence of different factors. Therefore, we can define translation as translation. It is the second step in the process of protein synthesis, and translation, it is the process of formation of polypeptide or protein according to the genetic information present on mRNA in the form of codons. अर्थात मेसेंजर RNA वर कोडॉन्सच्या स्वरूपात जी जेनेटिक इन्फॉर्मेशन आहे त्यानुसार प्रत्यक्ष पॉलीपेप्टाइड म्हणजेच प्रोटीन मॉलिक्यूल तयार होण्याच्या प्रक्रियेला ट्रान्सलेशन असं म्हटलं जातं. Or the translation can be defined as translation, it is the process by which the synthesis of polypeptide chain is guided by the genetic information present on messenger RNA in the form of codons. Now, first of all, we people have to study about the different factors or machinery required for the process of translation. सुरुवातीला आपल्याला या ठिकाणी ट्रान्सलेशन साठी आवश्यक असणारे जे वेगवेगळे घटक आहेत, अर्थात the machinery for translation that we have to study. Just now I told you that translation, it is the second step in the process of protein synthesis, and it is nothing but the process during which actual polypeptide molecule or protein molecule is synthesized according to the genetic information present on mRNA in the form of codons. Now the machinery required for process of translation. First, amino acids. The amino acids acts as a raw material for the process of translation, for the process of synthesis of polypeptide or protein molecule, because we know that proteins are nothing but the polymer of amino acids. Proteins are nothing but long chain of amino acid. It means that the amino acids acts as the building block or monomer of proteins, and that's why the amino acids are considered as a raw material for the process of synthesis of protein or polypeptide. Now actually in the cells of living organism, for the process of synthesis of protein, in all 20 different types of amino acids are required. Protein याचा अर्थ long chain of amino acid, म्हणजेच प्रोटीन तयार होण्यासाठी वेगवेगळ्या प्रकारच्या अमायनो ऍसिडची गरज असते आणि अमायनो ऍसिडला म्हणून प्रोटीनचं बिल्डिंग ब्लॉक मोनोमर किंवा मूलभूत एकक असं मानलं जातं. सजीवांच्या पेशींमध्ये प्रोटीन्स तयार होण्यासाठी 20 वेगवेगळ्या प्रकारच्या अमायनो ऍसिडची गरज असते and all these 20 different types of amino acids are present in the cytoplasmic matrix. It is also known as amino acid pool. So the first requirement for the process of protein synthesis is amino acid, which acts as the raw material. Second, DNA. DNA acts as a specificity control. As a specificity control. The process of protein synthesis, the process of translation is highly complicated, and the genetic information present on the DNA in code language, it directs or it guides the actual mechanism of synthesis of protein having the particular number of and particular sequence of amino acid. It means, in brief, the DNA, it guides, it directs the actual process of protein synthesis. It has a specificity control over the process of protein synthesis. अर्थात विशिष्ट प्रकारचं प्रोटीन तयार होण्याची जी माहिती आहे ती माहिती, ती इन्फॉर्मेशन ही डीएनए वर असते आणि किंबहुना त्या इन्फॉर्मेशन नुसार प्रत्यक्ष प्रोटीन मॉलिक्यूल हा तयार होणार असतो आणि म्हणून डीएनए हा संपूर्ण या प्रक्रियेवर एक स्पेसिफिसिटी कंट्रोल, विशिष्ट प्रकारचं नियंत्रण हे ठेवत असतो. नेक्स्ट, RNAs. All of you are known. In the process of translation or in the process of protein synthesis, all the three different types of non-genetic RNAs are involved, and among which the most important is mRNA. Protein synthesis च्या संपूर्ण प्रक्रियेमध्ये तिन्ही प्रकारचे non-genetic RNA हे इन्व्हॉल्व होत असतात, such as mRNA, rRNA and tRNA. But mRNA is most important because it carries the information from DNA up to the ribosome for the process of protein synthesis through transcription. कारण मेसेंजर RNA हा DNA वर असणारी इन्फॉर्मेशन रायबोसोम पर्यंत नेत असतो की जी प्रत्यक्ष प्रोटीन तयार होण्यासाठी गरजेची असते आणि म्हणून the mRNA acts as intermediate between the DNA and protein. Now come to the next, that is ribosomes. The most important factor for the process of translation. Ribosomes are nothing but these are tiny, very small ribonucleoprotein molecules. These are very small, tiny particles made up of ribonucleoproteins, and these are without membrane. These are non-membranous structure. Ribosomes are called as a protein factories. Why? Because the actual process of synthesis of protein or polypeptide is carried out with the help of ribosome. The ribosomes consist of two subunits. Structurally, ribosomes consist of two subunits, such as the larger subunit and smaller subunit. Larger and smaller subunit. These larger and smaller subunit remain separate from each other when they are not involved in the process of protein synthesis, and they come together, they join together only at the time of protein synthesis. It means the two subunits of ribosome remain dissociated, separate from each other when not involved in protein synthesis, not involved in translation, and are associated during the process of protein synthesis. Now, the ribosomes are of again two types. In prokaryotes, 70S, and in eukaryotes, 80S, isn't it? Let me write here. In prokaryotes, the ribosomes are of 70S type, and in eukaryotes, 80S type. In prokaryotes, the 70S ribosome has two subunits, and that two subunits are represented as larger one 50S and smaller one 30S. Please mind well, here this S stands for Swedberg unit or sedimentation coefficient. These figures or these values of the larger and smaller subunits are not additive. त्यांची बेरीज केली जात नाही. That sedimentation coefficient is depend upon the size of that subunit, size of that particle. Anyway, so the larger subunit in prokaryotic ribosome is 50S and the smaller subunit in prokaryotic ribosome is 30S, while the larger subunit in eukaryotes is 60S and smaller subunit as 40S. The ribosome has different binding sites. I am going to show by drawing diagram. या ठिकाणी मी डायग्राम कडून तुम्हाला दाखवतोय की रायबोसोम वर वेगवेगळ्या प्रकारचे बाइंडिंग साइट्स असतात. प्लीज लुक हियर. I said just now, each ribosome has two subunits. I am going to show. Consider here is the larger subunit of ribosome and here is a smaller subunit of ribosome. The ribosome has different binding sites, such as first one, let me show. This is supposed to be the mRNA binding site. mRNA binding site. It means during translation, the mRNA binds ribosome at this region. Then the larger subunit of ribosome has three different binding sites for tRNA. For tRNA. Look at the diagram. This is A site. This is P site and this is E site. A site means amino-acyl site, or it may be known as acceptor site. Acceptor site. P stands for peptidal site. Peptidal site. It may be known as donor site. And E, it is recently discovered in bacterium E. coli. E stands for exit site. Exit site. Now, what is the role of all these three different sites that we are going to study very soon while studying the actual mechanism of translation. Now, in addition to the amino acids, DNA, RNAs, ribosome, the process of translation also requires so many different types of enzymes such as amino-acyl transferase as well as the peptidal transferase. It also requires some cofactors such as magnesium ion and ATP and GTP as a source of energy. Now, actually, let us start the mechanism of translation. आता प्रत्यक्ष ट्रान्सलेशनचं मेकॅनिझम आपण अभ्यासणार आहोत. So let us talk about it. Mechanism of translation. It involves two major steps. First, activation of amino acids. Activation of amino acids and formation of AA-tRNA complex. Here AA stands for amino-acyl tRNA complex. Just now we have studied amino acids acts as the building block or monomer of protein or polypeptide, and that's why all the different 20 types of amino acids present in the cytoplasmic matrix are inactivated. They are first of all activated in presence of ATP as a source of energy and enzyme amino-acyl transferase. सर्वप्रथम आत्ता जसं मी तुम्हाला म्हटलं की प्रोटीन सिंथेसिस साठी प्रत्यक्ष पॉलीपेप्टाइड किंवा प्रोटीन मॉलिक्यूल तयार होण्यासाठी आवश्यक असणारे जे 20 डिफरंट टाइप्सचे अमायनो ऍसिड सायटोप्लाझ्मिक मॅट्रिक्स मध्ये असतात यांना ऍक्टिव्हेट केलं जातं. मीन्स दे आर प्रिपेअर्ड, they are made ready to take actual part in the formation of polypeptide, and this activation of amino acids is carried out with the help of ATP and enzyme amino-acyl transferase as well as magnesium ion as a cofactor. Let me show in diagram. Consider here is the cytoplasm, cytoplasmic matrix, and all the different amino acids here are activated. These are supposed to be the different amino acids. They are activated, and immediately after the activation of amino acids, each amino acid bind with the 3' end, that is CCA end of specific tRNA. Look, these are supposed to be the tRNA molecules, and this tRNA molecule bind to the specific amino acid with its 3' end CCA end to form amino-acyl tRNA complex. Now all these are the amino-acyl tRNA complexes, that is AA-tRNA complexes are formed. This is the first step, first major step in the mechanism of translation. So the first step in the mechanism of translation is the activation of amino acid and formation of AA-tRNA complex. Now come to the second step, that is formation of polypeptide chain, polypeptide molecule. It involves three major steps such as initiation, elongation and termination. प्रत्यक्ष पॉलीपेप्टाइड चेन तयार होण्याची प्रक्रिया ही तीन भागांमध्ये विभागली जाते. इनिशिएशन, नेम itself indicate, initiation means the beginning. Elongation, that is the amino acids get added one by one so that there is a formation of polypeptide chain, there is an elongation of polypeptide molecule. And termination means the end of process of translation, end of synthesis of polypeptide chain. Let us talk first, that is initiation. Initiation, it is the first step in the process of protein synthesis. Initiation is the first step in the process of translation. It requires messenger RNA, larger and smaller subunits of ribosome, different types of AA-tRNA complexes, enzymes, and also it requires the ATP or GTP as a source of energy. Initially, the smaller subunit of ribosome binds at the 5' end of messenger RNA. Look at the diagram. Here is a messenger RNA. We know that its one end is 5', other 3', and it shows the sequence of nucleotides grouped in the form of codons. Here I am going to show the sequence of nitrogen bases or nucleotides, and the triplet of nucleotides is nothing but the codon. So I said that initially the smaller subunit of ribosome binds at the 5' end of messenger RNA to form mRNA, smaller subunit ribosome complex, and immediately it is followed by the association of larger subunit of ribosome. It is followed by the association or binding of larger subunit of ribosome to this smaller subunit and mRNA complex. We have already studied this larger subunit has three different sites such as A site, P site and E site. Going to show in diagram. Please look. This is A site, amino-acyl or acceptor site. Here is second P site and third E site, exit site. Now always at the 5' end of messenger RNA, first codon is in the form of AUG, very rarely GUG, and it codes for amino acid methionine in eukaryotes and N-formyl methionine in prokaryotes. Therefore, this AUG, it attracts the tRNA with complementary anticodon UAC and carrying amino acid methionine. You understand? मी आता असं म्हटलं की नेहमी मेसेंजर आरएनएच्या 5' एंडला असणारा पहिला कोडॉन, ज्याला आपण इनिशिएशन कोडॉन किंवा स्टार्ट कोडॉन असं म्हणतो, हा AUG असतो. काही वेळेला if AUG is not available then it is replaced by GUG, but it always codes for amino acid methionine in eukaryotes. पण हा AUG असो नाही तर GUG हा युकॅरियोटिक सेल्स मध्ये मिथ्योनिन या अमायनो ऍसिडला कोड करतो आणि प्रोकॅरियोट्स मध्ये एन फॉर्माइल मिथ्योनिनला कोड करतो. And therefore this AUG, it attracts the tRNA having anticodon, complementary anticodon to AUG, UAC, isn't it? UAC carrying amino acid methionine, carrying amino acid methionine, and this first AA-tRNA complex, this first AA-tRNA complex carrying amino acid methionine comes and occupies the P site directly, comes and occupies P site directly. Look, याचा अर्थ जो पहिला AA-tRNA कॉम्प्लेक्स मिथ्योनिन या अमायनो ऍसिडला घेऊन येतो तो सरळ या ठिकाणी लार्जर सब युनिट वर असलेलं जी P साइट आहे, पेप्टिडाइज साइट आहे, त्या ठिकाणी जाऊन बाइंड होत असतो. बट द नेक्स्ट न्यू कमिंग AA-tRNA complexes always initially occupies the A site and then shifted to the P site. Try to understand. याचा अर्थ याच्या नंतर जे नवीन AA-tRNA कॉम्प्लेक्सेस इकडे येणार आहेत ते मात्र सुरुवातीला ए साइटला ऑक्युपाय करतात, ए साइटला बाइंड करतात आणि नंतर ते ए साइट वरून पी साइट वर शिफ्ट होत असतात. This process is known as the initiation. It is the beginning of synthesis of polypeptide chain. It also requires some initiation factors. यासाठी काही initiation factors पण गरजेचे असतात. Initiation is followed by the second step, that is elongation. Elongation. Elongation is the process in which new AA-tRNA complexes are attracted toward the ribosome and thus the new amino acids get added to the previous one according to the sequence of codons present on mRNA. Elongation या दुसऱ्या स्टेप मध्ये आता नवीन AA-tRNA complexes राइबोसोम कडे अट्रॅक्ट होणार आहेत. Of course, according to the codons present on mRNA, mRNA वर असणाऱ्या codons नुसार AA-tRNA complexes इकडे अट्रॅक्ट होतील, म्हणजेच नवनवीन अमाइनो ऍसिड्स हे त्या ठिकाणी ऍड होत जाणार आहेत and hence there is an elongation of chain of amino acid, elongation of polypeptide chain. Now the second amino acid is brought by next AA-tRNA complex and it occupies the A site. It occupies the A site. Look at the diagram. आता या ठिकाणी दुसरा AA-tRNA कॉम्प्लेक्स राइबोसोम जवळ येईल आणि तो A साइटला बाइंड करेल, A साइटला ऑक्युपाय करेल. This second and first amino acid are far close to each other and therefore there is a formation of peptide linkage, peptide bond between the two adjacent amino acid in presence of enzyme peptidile transferase. अर्थात या ठिकाणी हा दुसरा जो AA-tRNA कॉम्प्लेक्स A साइट वर येऊन बाइंड झालेला आहे, त्याने आणलेला अमाइनो ऍसिड आणि फर्स्ट अमाइनो ऍसिड दॅट इज मिथिओनिन या दोघांमध्ये पेप्टाइड बॉन्ड तयार होतो इन प्रेझेन्स ऑफ एन्झाइम पेप्टिडाइल ट्रान्सफरेज. नाउ आफ्टर दिस द राइबोसोम मूव्स towards the 3' end by a distance of single codon only at a time. आता हा राइबोसोम 3' end कडे सरकतो by a distance of single codon, by a distance of 1 codon each a time, every time. प्रत्येक वेळेला राइबोसोम फक्त एक कोडॉनने पुढे सरकतो, पुढे याचा अर्थ 3' end कडे सरकतो. Now let me show in another diagram. Please look. As usual, 5' 3' end, first codon, second, third, fourth and so on. I said the ribosome moves towards the 3' end by single codon. I am going to show the previous position. As usual, here is the A site, P site and E site. As the ribosome moves towards the 3' end by single codon, then the tRNA present at P site, listen, tRNA present at P site becomes free and it is released from the ribosome via E site. It is released from the ribosome via E site. E stands for the exit. It is supposed to be a door through which this tRNA has to go out. जणू काही हे मागचं दार आहे ज्याच्याने या tRNA ला बाहेर निघून जायचंय. So I said that as the ribosome moves towards the 3' end by a single codon, the tRNA at P site becomes free. tRNA becomes free and it leaves the ribosome, while the tRNA present at A site occupies the P site. पर्यायाने A साइट वर असणारा जो tRNA आहे तो P साइट वर येतो and thus A site becomes vacant for new coming AA-tRNA. पर्यायाने आता A साइट ही वॅकंट झाली, रिकामी झाली. कोणासाठी? फॉर न्यू कमिंग AA-tRNA. परत नवीन येणाऱ्या AA-tRNA साठी A साइट वॅकंट झालेली आहे. And thus now continuously this process occurs towards the 3' end. New amino acids get added and there is a formation of chain, that is nothing but elongation of polypeptide chain. I am going to show here once again in one diagram. Please look. Now suppose this ribosome has reached near the 3' end. आता कल्पना करा या रायबोझोम या ठिकाणी 3' end पर्यंत आलेला आहे. And here is a formation of chain of amino acids, that is polypeptide molecule. As usual, I am going to show different sites, A, P and E site. Okay. After the process of elongation, as the ribosome reaches at the 3' end, at 3' end there is a stop codon. There is one of the nonsense codon. You know that nonsense codon may be in the form of UAA, UAG or UGA, UGA, and these nonsense codon do not code for any amino acid. Why? Because there is no complementary anticodon to these nonsense codon. या नॉनसेन्स कोडॉनला कॉम्प्लिमेंटरी अँटीकोडॉन नसतो आणि म्हणून हे कुठल्याही प्रकारचं अमायनो ऍसिडला कोड करत नाही. Therefore, as the ribosome reaches at the 3' end, there is no chance of new incoming AA-tRNA complex carrying new amino acid. पर्यायाने या ठिकाणी हा रायबोझोम 3' एंडला पोहोचल्यानंतर आता नवीन AA-tRNA कॉम्प्लेक्स नवीन अमायनो ऍसिडला घेऊन येण्याची शक्यता नाही. And therefore the ribosome moves forward. पर्यायाने रायबोझोम हा पुढे सरकतो and the polypeptide chain becomes free. Messenger RNA also becomes free and the two subunits of ribosome, that is larger and smaller subunit, dissociate from each other, get separated from each other, and that is the end of synthesis of polypeptide chain. Means it occurs the termination, that is termination of synthesis of polypeptide chain occurs, and it is the last step in this mechanism of translation. Now, after become free, the mRNA get degenerated quickly by the influence of enzyme nuclease, because mRNA is short-lived. So, immediately after the end of process of translation, the mRNA is degenerated by the influence of enzyme nuclease. Now, during the process of translation, there are certain part of messenger RNA which has not undergone the translation, such as a short part of mRNA before initiation codon, that is at the 5' end. You know, at the 5' end of messenger RNA there is a presence of some nucleotide sequence, methyl guanosine triphosphate. In the same manner, at 3' end, after termination codon, after stop codon, there is a adenine. So these nucleotide sequences present before initiation codon and after termination codon does not undergo translation, and therefore that sequences are called as untranslated region, UTR, untranslated region. That's all about the entire mechanism of translation. So let us stop here. Thank you.